Welcome to another exciting lecture from Mr. P. We're going to, in this lecture, talk about Chapter 7, but from a whole chapter perspective. So as I sit here and look at my agenda for the week, I see that you have a test coming up. And so in this lecture, I thought that I would kind of hit the highlights all in one lecture so that as you're studying for the review or studying the review for the exam, you don't have to flip around and look at all the individual lectures. While I definitely think that's beneficial, I wanted to create one that kind of synthesized a lot of the information into a very informationally dense review session for you. So we're going to start with the cell theory. If you remember, the cell theory is essentially one of those fundamental theories or fundamental ideas to the discipline of biology. In fact, there are three components to the cell theory. One is that all living things are made of cells. Okay? Cells are the basic unit of life. All living things are made of cells, and all cells come from existing cells. So you will remember that during the time when we were discussing this, we stated these three components over and over and over. Okay? Cells are the basic unit of life. Okay? Or cells are the basic unit of all organisms, which means that nothing in that organismal level or nothing within an organism is more basic than the cell. The cell is the basic unit. You could also talk about it in terms of the cell is kind of the monomer of an organism. Okay, if you remember from chapter 2, when we did all of the biochemistry stuff, we talked about monomers and polymers. We talked about monomers which are put together to produce polymers. Cells can be looked at as a monomer that when put together with other monomers, cells in this case, it would create an organism or a multicellular organism. Okay, an organism is made up of multi-cells, okay, or a bunch of cells, and so we consider them multicellular. So cells are the basic unit of life. All organisms are made up of one or more cell, and the reason it is one or more cell is because cells can live as single cellular organisms, is what I'm trying to say. So organisms like the ones you saw in lab, okay, we looked at paramecium, we looked at euglena, we looked at amoeba, we looked at your cheek cells, and while your cheek cells don't particularly live and do all of the things that cells do as single-celled organisms, those single-celled protists definitely do. So the paramecium are going to move around, they're going to consume, they're going to respond to the environment, they're going to do all those things that we talked about living things doing way back in chapter 1, but they live as a single-celled organism. Okay, they can definitely live as more than one cell, which is apparent when you look at you or any other animal. Okay, we are a multicellular organism made up of individual cells, which all have individual or unique specialized tasks, which when they come together, they produce an organism that is very complex like yourself. And then the final component is that all cells come from pre-existing cells. And if you remember, Virchow was the one that said that. And he specifically studied cell division and witnessed that when cells come to the end of their life cycle or come to the end of the cell cycle, they will divide and one cell will produce two, and those two cells will become four. And that is the basic premise for why organelles or organisms can grow and develop. Okay, so over here in this triangle we have the cell theory. You will see Schleiden, Schwann, and Virchow. There are two guys that we have not identified in this little triangle, and those two guys are Hooke and Leeuwenhoek. Okay, I know you can't read that very well, but Hooke and Leeuwenhoek. Hooke was the first one to identify cells, and he's actually the one that coined the term cell. Remember, he looked at cork. Leeuwenhoek came along after the fact and made the microscope better, as well as looked at pond water, and he was the first to identify living cells. So he was the first to identify real-life living cells. He called them animalcules. Those animalcules were basically coined animalcules because they resembled tiny animals or microscopic animals. But the three kind of most important contributors to the cell theory, which is why they're actually in this pyramid to begin with, Schleiden is the one that said all plants are made of cells. Okay, so if I extend that arrow over here, he's the one that looked at cells within plant tissue and was able to establish that section of the cell theory. And after several trials of looking at plant material, he basically concluded that every single one of the cells or every single one of the plants that he looked at all contained these cells. And you looked at Elodia in the lab, and you should have seen something that resembled this. Notice all these tiny little green discs. All of those are chloroplasts, which are one of the organelles that you are tasked with knowing. Okay, those are the photosynthetic organelles. They're the ones that turn light energy into chemical energy, which is a form of energy the cell can use. But all plants are made of cells. Schleiden is the one that said that. Schwan. Remember I said you can remember Schwan because it's a majestic goose, right? Schwan is the one that said all animals are made of cells. And so he looked at tissues from various animals and concluded that, in fact, every single animal tissue he looked at all contained cells. Okay, so Schwan, majestic goose. Goose is an animal. All animals are made of cells. Schleiden, or Schleiden, is the one that looked at plants. He concluded that all plants are made of cells. You can remember that because you slide down a slide into wood chips. And wood chips are made of plant material, right? If you looked at a piece of wood chip under a microscope, you would in fact see all of these cells just like you do in a living plant, because wood chips are wood and wood is plant. Virchow, he's the one that looked, like I said before, he's the one that came up with this component, right? He looked at cell division and basically concluded that all cells come from pre-existing cells, and he found that out on multiple occasions. Okay, so that's cell theory and the contributions of those various individuals on the cell theory. Know that stuff. Okay, now let's change pace a little bit and get into the differences and similarities of prokaryotic versus eukaryotic cells. Okay, first, let's go through the both. All cells, okay, and so I'm saying both, but I'm pretty much saying all cells contain DNA or some kind of genetic material, most case DNA, and so we'll just say all cells contain DNA. All cells will contain chromosomes. All cells will contain ribosomes. However, we noticed or noted, and I'm just going to do a difference because we are on the ribosome talk, that prokaryotes, remember, contain 70S ribosomes, while eukaryotes contain 80S ribosomes. That means nothing to us at this point in the class other than the 80S ribosomes are larger and the 70S ribosomes are smaller. Think about prokaryotic cells are smaller anyway. Okay, they're typically much smaller. Eukaryotes are much larger, so it makes sense that 80S would be a larger ribosome within a larger cell, and a 70S ribosome is a smaller ribosome within a smaller cell. All cells contain cytoplasm. All cells contain a plasma membrane. Sometimes both cells will contain cell walls. Remember, all prokaryotes have a cell wall. All plant cells have a cell wall, which would be within this eukaryote region, but not animals, or animal cells do not contain a cell wall. So you can't say all cells have a cell wall, but sometimes both of them will have cell walls. And then vacuoles. All cells have vacuoles. However, again, there's a difference between plant and animal cells when it comes to the vacuole. The vacuole is much larger on a plant cell, much smaller on an animal cell. Okay, let's talk about, so both cells are identified as having some kind of DNA as a genetic material. We know that DNA is kept within the nucleus of a eukaryotic cell and within a nucleoid region of a prokaryotic cell. The DNA is linear in shape, okay, and so the DNA will actually resemble an X-shaped chromosome on the eukaryotes. The prokaryotic chromosomes are actually going to resemble a ring. We call that a plasmid, okay, plasmid, but it's a circular DNA, and it floats freely around the cell. The nucleoid contains simpler DNA within a prokaryote, and the nucleus contains complex DNA within the eukaryote. Eukaryotes have membrane-bound organelles. The prokaryote has no membrane-bound organelles. Okay, there are organelles within the prokaryote, but the ribosome is kind of looked at as the only organelle within the prokaryote. Eukaryotes will divide by mitosis and meiosis. Those are both forms of cell division. Prokaryotes will divide by binary fission. Prokaryotes are typically always unicellular. Remember, prokaryotes are actually bacteria, or I should say bacteria are examples of prokaryotes. Eukaryotes would be things like animals and plants. Okay, so these would be eukaryotes. Bacteria would be prokaryotes. It makes sense that animals and plants would be eukaryotic, while bacteria are prokaryotic, because prokaryotes are always found, or typically always found, in a unicellular form. And plants and animals are always multicellular and a lot more complex. Eukaryote are most of the time aerobic, and prokaryotes can actually be anaerobic or aerobic, and that'll make more sense when we get into the cell respiration in Chapter 9. Okay. There is a question on the test that has you compare and contrast prokaryote, eukaryote. Where does that information come? Probably from this guy right here, this chart. Let's look at the prokaryote, eukaryote from a pictorial representation. Now we have a prokaryote over here. We have a eukaryote over here. Notice that there is a complexity difference. Okay, so if we start with the prokaryote, things that I think you need to identify is that there is no nucleus here. See, there's just a bunch of DNA that is free floating in a nucleoid region. All of that DNA is going to be kind of encapsulated by all these little dots, and you'll see these little dots down here. All those dots are ribosomes, and in fact those will be the 70S ribosomes. Their job, sole job, is to read the DNA and essentially produce proteins that that cell can use. And that's not just a prokaryotic ribosome function, that is a ribosome function across the board. We also have flagella. Remember, flagella and cilia are both modes of transport for a cell. Flagella is actually a single whip-like microtubule projection that is going to be used as a whip-like propeller to transport this cell through its environment. These pili look like look like cilia, but they're not. Okay, pilia are typically for attachment. Okay, attachment, while cilia is for the movement of materials around the outside. Okay, around the outside of this cell, you will have a basically a cell wall. Okay, there's a cell wall made up of peptidoglycan. Sometimes you'll have a capsule. You'll always have a cell wall. You'll always have an actual membrane or cell membrane, and then you'll have cytoplasm on the inside of the membrane that just makes up the volume of the cell. Okay, very simple, very small, very simple. The only thing you really have is a nucleoid region that contains DNA that is not surrounded by a nucleus. You'll have ribosomes to make proteins. You'll have a membrane. You'll have a cell wall, and sometimes you'll have a capsule. And then you'll have a way to either attach it to something in its environment or move it around its environment. Okay, if we contrast that very simple prokaryotic cell with a eukaryotic cell, you'll see that you have an actual nucleus. That nucleus will contain a core called the nucleolus. That core contains most of the DNA. Okay, the DNA is going to be in a tight core called the nucleus or the nucleolus. That nucleus contains a double membraned nuclear envelope. Okay, and that nuclear envelope will have pores. Those pores allow for RNA messages and ribosomes to be created in the nucleolus. They will leave the nucleus through one of those pores and then will essentially be synthesized by other cytocomponents, okay, or cell components in order to do specific jobs. But ribosomes and RNA messages can be created in the nucleolus and then will leave through a nuclear pore. If it's a ribosome, it's going to either attach to the rough ER, and you'll see all of this kind of interconnected membrane system. That's the rough ER. Notice all those little ribosomes that are attached. Those ribosomes are going to stud this membrane, which gives it the appearance of being rough. Okay, it's a rough endoplasmic reticulum. Next to the rough ER will be the smooth endoplasmic reticulum. It's all out here on the outside. Notice that it does not contain ribosomes. Its job, the smooth ER's job, is to synthesize and modify lipids. The rough ER's job is to house proteins or house ribosomes and modify proteins. Okay, but it's specifically the job of ribosomes to produce the proteins. The rough ER kind of just helps modify the proteins. Mitochondria is an organelle that's in charge of producing energy. Sometimes you will see chloroplasts if it's a plant cell. This is an animal cell, so you will not see chloroplasts. You'll also not see a large central vacuole. Cytoskeleton is a way, just like your skeleton, to create structure and support of the cell from inside. It gives the cell its shape. It gives it its structure. It doesn't allow it to kind of crumble on itself. Golgi apparatus. Golgi apparatus is this guy. He's sitting right there in the cytoplasm, and his job is to receive proteins that come from the rough ER via a vesicle, produce a new vesicle which will either embed itself in the membrane to export the protein to outside or the extracellular components of that cell, or will stay in the cell and produce a lysosome, which can be seen in here. It's not labeled. One of those little vesicles is going to produce or house digestive enzymes. We call that lysosomes. Centrioles are in charge of cell division. We're going to learn more about that in chapter 10 when we do mitosis and meiosis. And then you'll have a plasma membrane or a cell membrane around the entire outside of the cell, which its sole job is to serve as a boundary between the inside and outside, and also regulate what goes in and out of the cell. Okay, so that's prokaryote, eukaryote. Also questions about that on the test. Now, let's go through the components of the cell membrane. Okay, components of the cell membrane. You've got the main component of the cell membrane, and so when I say main component of the cell membrane, I would highly encourage you or expect you to say phospholipid. Okay, so we've got all of these phospholipids. You know from chapter 2 that phospholipids have a head and a tail. In fact, they have two tails, right? So this is a bilayer of phospholipids. I would also expect you to be able to draw this on the test. Okay, those heads are polar and hydrophilic. The tails are non-polar and hydrophobic. Okay, hydrophobic tails are going to repel water. Hydrophilic heads are going to attract water. So water is going to be outside of the cell, water is going to be inside of the cell, but water will not go into the membrane. One thing that gives the structural integrity to the membrane are these random cholesterol molecules that are embedded in the membrane. Okay, so I'm going to label these cholesterol. Okay, cholesterol are the, essentially is the mortar between bricks that hold a brick wall or a stone wall together. Okay, they give the structural integrity to the membrane. Next to the phospholipids that span the entire membrane will be proteins. Okay, and so if this protein spans the entire membrane, it is called an integral protein. Okay, because it is basically completely embedded in the membrane. This one happens to be a channel protein. Okay, channel proteins provide a channel by which materials can move into and out of the cell. Channel proteins are specific to the substrates that they move. And that's all you really need to know about that. Okay, then next to that we will have more phospholipids. Okay, because remember, phospholipids make up the main component of the cell membrane. We also can have peripheral proteins. Okay, so this peripheral protein is essentially not embedded in the entire membrane. It is sitting there on one side. It can either be on the outside or it can be on the inside. This is a, like I said, peripheral protein. This is a channel protein, also called an integral protein. We also have the another component, which is an integral protein but has a long carbohydrate chain on one side. This is called a glycoprotein. Okay, glyco for sugar, protein for protein. The protein is the protein component that is actually embedded in the membrane. Glyco is sugar or carbohydrate, so it is the actual carbohydrate component of that protein. That glycoprotein or the carbohydrate serves as a name tag for the cell. Okay, name tag. So it helps the cell identify itself. It helps the cell tell other cells that are neighboring it what kind of cell it is. And this makes sense because all of your organ tissues touch each other. Your heart touches your lungs touch your liver. Okay, if the liver cells can't communicate with the lung cells, they might just grow together, or they might send products that each other doesn't need or each other doesn't want, right? So they've got to be able to basically communicate with each other, and so that's the purpose of these glycoproteins. Now, because we have a membrane that serves as a boundary and regulates what goes into and out of a cell, it gives rise to cell transport. Okay, it's really, really important for cells to be able to transport materials to and from the cell. Okay, there is a distinguish between passive transport and active transport. Active transport requires energy, and that energy is always or typically always in the form of ATP. ATP, as we will learn next chapter in chapter 8, adenosine triphosphate. You will learn that molecule well next chapter. But for now, you just need to know ATP is the energy source for a cell. Active transport requires the addition of energy to move things against the concentration gradient. Okay, against concentration gradient. Passive transport moves materials with a concentration gradient. Now, what is a concentration gradient? Concentration gradient is high to low. So think about elevation. Does it require you any energy to roll down a hill? Okay, if we were to roll down this hill from high to low elevation, you just let gravity take over and it does it for you. You don't have to actually put any energy. You don't have to hike down the hill. You just let gravity pull you down the hill. Okay, now if you were to switch that and go up a hill from low to high, that requires you not to just sit down and let gravity take you, but you have to physically do work to get up a hill. Same thing with molecules. Okay, if we have a high concentration here and a very low concentration here, things will naturally diffuse from high to low through a membrane. That is diffusion. Okay, diffusion. You can see diffusion by dropping food coloring into water or by spraying a fragrance like cashmere woods into a room, right? Eventually the room will fill up with fragrance because that fragrance will diffuse from an area of really high concentration where it was sprayed to a really low concentration where the fragrance isn't at, and so it'll eventually diffuse. That does not require a membrane, but diffusion can happen through a membrane. Let's talk about facilitated diffusion. You will see that it is still high to low, just like this one. Okay, the only addition or the only difference is the addition of this protein. This is a channel protein. It provides a channel by which those solutes move through this membrane. That channel protein is very specific to the solutes that are moving through. However, it doesn't require energy. It's still passive. It's still high from an area of high to low. Okay, so it's going to move down a concentration gradient from high to low without energy. Still passive, but it moves through a channel protein. It is helped through a protein, and so we call it facilitated diffusion. Okay, now the third one, let's talk about active transport. Active transport, like we said before, requires the addition of energy. You'll notice that this is the low energy. This is the low concentration. This is the high concentration. And so when solutes move from low to high, they have to go against the concentration. And so they have to, just like going up this hill, they have to impart energy in order to get there. Okay, active versus passive, very, very important. Osmosis is a form of diffusion, so it is a form of passive transport. Very, very important for you to know that it is passive transport. Osmosis, by definition, is the diffusion of water from an area of high water concentration. Okay, if I put these in parentheses, it's an area of high water concentration to an area of low water concentration. Now we can do the opposite, and we can say osmosis is the diffusion of water from an area of high solute, okay, well, I should say backwards, from an area of low solute to an area of high solute, because you know that if you have low solute, by default, you have to have a larger solvent, right, or water in this case. If you have a high solute, like salt, then you have a lower solvent, or water. So if we just kind of throw the solute out, like I did in this equation, and we just say it goes from high water to low water, you will always be right. So let's do an example problem. Okay, if I get my eraser, we can erase this. And let's do an osmosis problem or an osmosis practice. Okay, you will have to do an osmosis practice on the test. Probably have to draw something similar to this. And so let's just assume that we placed this starch weenie, okay, and it should look similar to what you did in the lab. If we did something that looks like this, it's probably very familiar. You put a solution inside the starch weenie or inside the dialysis tubing. We used NaCl. It was a salt solution, and so let's assume we put a 15% NaCl solution in the bag. We also have water in the bag because we dissolved the NaCl in water. Okay, so let's assume we have, if we have 15% NaCl in the bag, then we probably have 85% water, which makes up the difference between the 15 and 100%, right? So 85%. On the outside of the bag, let's assume we put 95% H2O in the environment, and we also put 5% NaCl outside the starch weenie in this environment. Okay. Now, if the membrane is permeable to water, water is going to want to diffuse through the membrane. Water is going to always go high water to low water. So water is going to go from an area of 95% to an area of 85%. Water will transport through the membrane and will penetrate into that dialysis tube bag. And so it's going to swell. Water molecules are going to come in here and it's going to fill up. So you would expect the mass to increase in that starch weaning over a given amount of time. If the bag was permeable to salt, 5% versus 15%, you would expect salt to move out. Both of these are wanting to reach this fancy term called equilibrium. Okay, the salt's moving out because it wants to mix in the middle between 15 and 5%. It probably will mix at about 10%. Once the outside and the inside of this cell is 10%, they are considered isotonic, or the solutions would be considered isotonic. They would have reached equilibrium at that point. Water, 85 and 95, probably wants to mix at about 90%, right? Because that's somewhere between 95 and 85, so 90% water. Again, that'll be isotonic with the water concentrations that's inside that cell. Okay. If we throw in some other terms, this bag originally was hypotonic. Well, scratch that. It was hypertonic because we had a higher concentration of solute, meaning hypertonic. The outside of the bag would be hypotonic because it had a higher water concentration. Hypo is lower solute, hyper is higher solute. And those are terms that are just used to essentially look at the difference between two environments or two kind of concentrations. Okay. Now, what happens if we put red blood cells into a solution? Okay, and this has medical implications, right? You don't want to give somebody, if somebody's dehydrated, you don't want to just like inject a syringe full of pure water. Why? We'll talk about it. In an isotonic solution, any cell, in this case red blood cell, will have a net movement of water which is 0, okay, or a 0 net movement. That means because the concentration on the outside is the same as the concentration of the solute on the inside, you're going to have just as many water molecules moving out as you do moving in. Okay, so if it's the same, for every one molecule that moves out, one molecule moves in. And so your net movement is 0, and so the mass and the volume of these cells remains the same. In a hypotonic solution, okay, that means that, let's say the red blood cell is 90% water and the environment is 100% water, because hypotonic would mean that the environment is less solute and higher water or higher solvent. So if there's 100% of water on the outside and only 90% of water on the inside, water is going to go in, causing the cells to swell, and eventually, just like a water balloon taking on too much water, or you try to overfill a water balloon, they're going to eventually rupture. Why would injecting somebody with 100% water be bad? Your red blood cells would literally explode. Okay, that's bad news. You don't want to see exploding red blood cells because red blood cells that have exploded are not very good at transporting oxygen, which is what red blood cells are intended to do. Okay, structure dictates function. We've said that over and over. Red blood cells that are ruptured are not very good at, or not structured well in transporting oxygen. Now the opposite, hypertonic. What happens if you put your red blood cells into a 50% water solution? Okay, 50% water solution. Like what happens if you inject salt water from, or even drink salt water? There's a reason people always tell you not to drink the ocean, okay, or drink salt water. It's because you're putting a hypertonic solution into your body. You are bathing your own body cells in a hypertonic solution. So what happens? Because your red blood cells are actually higher water concentrated, or have a higher water concentration than the outside seawater, the cells want to actually lose water because they are trying to go from high to low again. They're higher concentrated in the salt, or in the red blood cell, lower concentrated in the environment, and so water is actually going to be pulled out of your cells, causing the red blood cells to shrivel up, which is also, like this case, very bad because, again, structure dictates function. These cells are not going to be structured well enough to transport oxygen, and probably you'll die. Anyway, that is an actual real-world application of osmosis. It is very, very important for you to know that water always wants to travel from an area of high water to an area of low water. As long as you can identify what part of a problem or what part of the environment is higher water and what part is lower water, you're going to be totally fine. Okay? I know there are some other things that may be on the test. This video is not intended to be all-inclusive of the test, but I think it'll do a great job in this 30 minutes. If you really understand what was talked about, I think you'll do pretty well on the test. Okay? If there are other questions, make sure you ask your teacher. But until then, good luck studying. Good luck on the test. Mr. P, out.